Hello there everybody, and welcome back to the channel. Today we're going to explore the fascinating processes of DNA replication, transcription, and finally translation, as well as outline what might happen if several different mutations occurred. DNA, or deoxyribonucleic acid, is the genetic material that carries out the instructions for building and maintaining an organism. It consists of nucleotides, which are made up of three key components: deoxyribose sugar, a phosphate group, and a nitrogenous base. Let's take a closer look at these components. Deoxyribose is a 5-carbon sugar that forms the backbone of the DNA molecule. The phosphate group links these deoxyribose sugars together to create the structure of a DNA strand. The nitrogenous bases are adenine, thymine, guanine, and cytosine. These bases are crucial because pyrimidines and purines only pair with each other. In DNA replication, several enzymes play crucial roles. To begin, we have helicase. Helicase is responsible for unwinding and unzipping the double-stranded DNA molecule, creating two single strands to serve as templates for replication. Topoisomerase helps relieve the tension created by the unwinding process. It prevents overwinding or tangling of the DNA strands. Next, we have primase. Primase synthesizes short RNA sequences known as RNA primers. These primers are essential because DNA polymerase requires an existing strand to start building a new DNA strand. DNA polymerase III is responsible for adding complementary nucleotides to the single-stranded template, forming a new DNA strand. It can only add nucleotides in the 5-3 direction. Okazaki fragments on the lagging strand need to be joined together, which is where ligase comes into play, sealing the nicks between the fragments, resulting in a continuous double-stranded DNA molecule. DNA polymerase I also has a role in DNA replication. It helps remove the RNA primers and replace them with DNA nucleotides. DNA replication occurs in the nucleus of eukaryotes and cytoplasm of prokaryotes. In the cell, DNA contains the genetic instructions needed to build and maintain an organism. To carry out these instructions, cells need to make RNA, which acts as a messenger molecule. RNA, or ribonucleic acid, is composed of a ribose sugar, phosphate group, and one of the 4 nitrogenous bases: adenine, uracil instead of thymine, cytosine, or guanine. Now, let's introduce the 2 key players in the transcription process: helicase and RNA polymerase. Helicase, like in replication, is the enzyme responsible for unwinding the DNA double helix like a zipper unzipping. Next, we have RNA polymerase, another essential enzyme. RNA polymerase is responsible for reading the DNA template strand and synthesizing the complementary RNA strand. The transcription process begins with helicase unzipping the DNA double helix at the specific region where RNA synthesis will occur. Once the DNA is unzipped, RNA polymerase binds to the promoter region, a signal that tells it where to start transcription. As RNA polymerase moves along the DNA template strand, it adds complementary RNA nucleotides. Remember, RNA contains uracil instead of thymine. A growing RNA strand emerges, following the DNA template. RNA polymerase continues along the DNA until eventually it reaches a termination signal, indicating the end of the gene, and transcription halts. The newly formed RNA molecule is released, and the DNA double helix zips back up. We've arrived at translation, a crucial step in protein synthesis, with 3 main steps: initiation, elongation, and termination. Let's begin with the initiation phase of translation. In both prokaryotic and eukaryotic cells, translation starts with the binding of a small ribosome subunit to the mRNA. Both the small and large subunit are made up of rRNA. Next, we move on to the elongation phase. In this stage, transfer RNA, or tRNA, bring amino acids to the ribosome. guided by the codons on the mRNA. Peptide bonds form between the amino acids, creating a growing polypeptide chain. Finally, in the termination phase, translation ends when a stop codon is reached on the mRNA. Release factors in the ribosome recognize the stop codon and trigger the release of the completed polypeptide. In both prokaryotic and eukaryotic cells, this process takes place in the cytoplasm as well as transcription. Finally, we've reached the mutations. It is important to understand that RNA is read and translated in groups of 3 nucleotides, also called codons. The first type of mutation that we have here is a substitution mutation, where we're substituting the A at the end of this codon for a C. Now, in this case, this mutation is invisible because both UCC and UCA create the same amino acid. Next is a deletion mutation. We're going to delete these 3 nucleotides, leaving us with this sequence. Now, this does not cause any substantial change in the overall sequence, and it only gets rid of 1 amino acid. Finally is an insertion mutation. Just by adding this nucleotide, this drastically changes the amino acid sequence and polypeptide chain that is created. And it is known as a frame shift mutation because the reading frame has changed and all of the bases pair differently. Instead of these 3, and these 3, it's these 3, these 3, these 3, and an extra nucleotide at the end, which creates a completely different sequence. That's everything I have for you today, and thank you guys for listening.